Good evening. My name's James Daveson. I'm a gastroenterologist. Welcome to this celiac disease webinar series hosted by Wesley Research Institute. This is the second of our series. The first one we had several months ago, which seemed quite popular. The series is really designed to enable the community to understand what's happening globally in terms of the advancement of therapies in the treatment of celiac disease, and also to provide, in conjunction with that, clinical updates on celiac disease, which we try to do together. I'll just move on to the acknowledgement of country. We'd like to acknowledge the Turrbal and Yuggera peoples, the traditional custodians of the land where Wesley Research Institute now stands, and we pay our respects to elders past, present, and emerging. Wesley Research Institute is Queensland's second oldest research organisation. It's located at the Wesley Hospital in Auchenflower. Wesley Research Institute conducts high quality ethical research across its multiple research programs, including the Clinical Trial Centre, WRI Biobank and its collaborators. It's a not-for-profit and it undertakes medical research to improve patient care. If we can just move on to the next slide. We are very grateful to our partners, including the Rosalie Martin event series. The Rosalie Martin event series was established in memory of Rosalie Martin AM, a former Wesley Research Institute board member, thanks to a generous gift from her family. This series, the Rosalie Martin event series, allows us to hold and conduct these webinars and to engage with the community and promote information related to celiac disease in this case. So we're very grateful to that family, and I've been very lucky to have met some of them. We'll just move on to the next slide. So tonight we're talking about celiac disease and genetics, about what families should know. And the first part will be from Dr. Richard Muir, and the second part by Dr. Paul Wagner, will speak about in a second. Myself, I'm a gastroenterologist. I'm clinically based. I do engage in research, both in Brisbane here and also regionally in Queensland. Richard Muir is a paediatric gastroenterologist. He's based at the Wesley Hospital here with myself. He sees a large number of patients with disease, and he also sees patients with other conditions such as inflammatory bowel disease, Crohn's disease, and ulcerative colitis. We've collaborated previously on research projects, and I'm very grateful that he's agreed to speak to us tonight. Richard will be providing us with an update of celiac disease in children. And finally, I'm very excited to have Dr. Paul Wagner join us as well. Paul's flown over from the United States to be with us. He's the Chief Executive of Forte Biosciences. It's a clinical stage biopharmaceutical company focused on autoimmune disease. He'll share his insights into their latest research and advancements in celiac disease, and perhaps offer a global perspective on emerging therapeutic approaches. So with that said, we might now move on to the first presentation for the evening, which is from Dr. Richard Muir. So thank you, Richard.
Sorry about that. My microphone wasn't working. So thanks very much for that introduction, James. Greatly appreciated for those kind words. So thank you as well to Wesley Research Institute for arranging the celiac webinar series. And also thank you to all the online attendees for the interest in tonight's meeting. So tonight I'll be talking to you a little bit about paediatric celiac disease. And it's important we talk about celiac disease in general, that it's a global disease. About 1.4% of the world's population will have positive celiac serology, what we call seroprevalence. About 0.7% will have biopsy-confirmed celiac disease. There is a reason for those difference in those numbers, and obviously access to healthcare. And sometimes we do see false positive markers with the celiac serology, but it's important to recognise quite a significant proportion of the world's population are affected. Some of the highest rates we see are in Europe and the Oceania region. In Australia, generally 1.25 to 1.4% prevalence, so a significant percentage of the population affected when you consider a population of 27 million people. There's a female to male ratio of 1.5 to 1, twice as common is in children as in adults. An important thing to think about as well is that in developed countries, around 80% of people with celiac disease remain undiagnosed, and it's higher in developing countries. And that has really important prognostic factors when you think about long-term unrecognized celiac disease, potentially comorbidities, health costs, etc. So a bit of a wordy slide, but what is celiac disease? It's an immune-mediated systemic disorder. We sometimes get focused on the gastrointestinal symptoms, but there's a number of other systems involved in the body, including the endocrine system and also musculoskeletal, to name a few. It's listed by wheat, gluten, and related grains, barley and rye in genetically susceptible individuals. We know it's characterized by the presence of a variable combination of gluten-dependent clinical manifestations, so GI and non-GI symptoms. And also just being aware, up to 30% of people with celiac disease will be asymptomatic at diagnosis. So certainly the absence of symptoms doesn't preclude you from a diagnosis of celiac disease. We know we have particular specific celiac disease-specific antibodies, and they're quite useful for screening. In certain situations in the paediatric population, they're useful for diagnosis in certain situations as well. We know that people with celiac disease have very particular genes, HLA or human leukocyte antigen DQ2 or HLA-DQ8. Take a step back quickly. A gene is basically a short segment of DNA which codes for a particular structure or function in your body. So these genes basically code for a structure we find on your white cells, and they help interact with other white cells as well, but we'll talk about that in a little bit. And celiac disease is characterized by the presence of enteropathy or damage to the small bowel. If I could summarize the genetics of celiac disease in one word, complex. Basically, we know there's a strong hereditary component. We see that celiac disease clusters in families. Also, we know, say, if you've got an identical twin, if one twin has celiac disease, there's about an 80% chance the other twin will develop celiac disease. So we have identified various genetic factors, and so HLA is the one which we know that all celiacs basically carry, the HLA genes. But the HLA genes are also found, DQ2 and DQ8, in the general population, and not everyone, obviously, in the general population develops celiac disease. So we know there are non-HLA genes involved. At the moment, there's probably about 50 which have been identified. There's also, to complicate the facts a little bit further, there's non-genetic factors. So we know that environment plays a role. So for example, obviously you have to have gluten to develop celiac disease, but there are other environmental factors that play a role as well. An evolving field is epigenetics, which is basically looking at how your environment, behaviors, lifestyle, potentially culture, actually affect the way that your genes are regulated and what we call expressed. So as we mentioned, celiac disease, HLA, 99% of patients with celiac disease patients will be positive for the HLA, DQ2 and DQ8, but around 50% of the general population also carry those genes. So if you're a member of the general population who just tests positive for that gene and you've got no first degree family member with celiac disease, your risk of developing celiac disease is about 2 to 3%. It's much higher, obviously, if you have a positive haplotype, a positive gene, and you have a first degree family member with celiac disease. We keep using a word, but what exactly is HLA? So easiest way to point it out is HLA is basically the structure we find on certain types of white cells. So an antigen-presenting cell is a cell in your immune system whose job and role basically is to sample its microenvironment. Basically what it's doing, it's gobbling up little different pieces of protein around it. So say viruses, bits of bacteria, etc. Or in this situation, a piece of gluten, a gluten breakdown product or a gluten peptide, and expressing that on its cell surface. And the structure which it expresses that cell on its cell surface is the structure which is coded for by those genes, HLA, DQ2, DQ8. And that in turn interacts with another cell in your immune system called a T cell. A T cell is kind of like a regulator cell. It controls a lot of other cells in your immune system, and it plays a key role in the development of celiac disease. Once that T cell is activated, it interacts with other cells via other receptors, but also it releases what we call cytokines, which are chemical messengers, which basically tell other cells what to do and switch them on and off. Obviously, in celiac disease, it's a little bit more complicated than that, and it's probably beyond the realms of today's talk to talk about the development of celiac disease. But I encourage you to look at a previous webinar where Dr. Daveson discussed the development of celiac disease and what we call the pathophysiology, or how it occurs. In short terms, so basically, gluten is relatively indigestible. It's broken down into some building blocks, what we call peptides, which are little clusters of amino acids that crosses into your intestinal wall, small bowel, and it's processed further by a number of proteins. And then that little cell, the antigen presenting cell we talked about before, it shows it on its cell surface and then shows it to the T cell, which then becomes activated. Obviously, there are a lot more cells involved in this. It's not as simple as that. And again, this is mentioning the fact that cytokines play a key role in not only the development of celiac disease, but also some of the complications we see with celiac disease. One more word about HLA. Not every HLA is created equal. So this is a nice little study. It's old, but it looks at people with celiac disease and looks at their children. So we know that those children will be at much higher risk of developing celiac disease, and they follow them up over time. So what you're looking at here is the rate of people with celiac disease, and then here is time. And these four separate lines are people with celiac disease with different haplotypes or different types of HLA. And you can see this bottom line here, they develop celiac disease at much higher rates than those people with this in this top line. So this bottom line is DQ2, up the top is DQ8. So just showing you that certainly some haplotypes we would consider higher risk for the development of celiac disease. Why is that? Basically, probably because the structure they code for, which is on the cell, on the antigen presenting cell, probably binds to gluten a little bit more effectively and presents it more effectively to your immune system. When you look at the geographical distribution of celiac disease, it brings up some interesting points. So if you look at, we said it's a global disease. North America, Mexico rates are similar to some Australian rates as well. But if you look at India, for example, in the north of India, around 1% have celiac disease. In the south, about 0.1%. That's despite them having quite similar HLA genes. So it's not the HLA which is causing the issue. There's obviously some other environmental factor which is playing a role. There are some follow-up studies looking at what happens when people from the south move to the north. We know that people in the north have more gluten than in the south, so that potentially that's one reason. But it'll be interesting to see what longer-term population studies show. So we sometimes get asked, well, okay, environment's playing a role. Is there anything we can do to reduce our risk? What can we change? We used to think that breastfeeding was protective, but although it has many, many benefits, obviously, it's not protective for the development of celiac disease. The one signal we've seen is that certainly high levels of gluten intake at introduction. So this is when a child's 4 to 6 months of age and starting solids. If they have high levels of introduction, particularly amongst those children who are first-degree relatives of people with celiac disease, as in son or daughter, that will increase your risk of celiac disease. Unfortunately, we can't come out and say, well, okay, this amount of grams will increase your risk and this amount of grams of gluten won't. But certainly some of the data coming from Sweden, some other places, they were quite high intakes of gluten. Like we're talking about the equivalent of a slice of bread, in a 10 to 12-month-old, which is really probably higher than you would see in a Western population, in Australia, sorry, for example. There's no difference between method of delivery. So cesarean section, but a vaginal delivery, there's no difference if your rates of the development of celiac disease. There is a potential association with early antibiotic use. So we think that that's probably going to be more reflective of the fact that you've exposed to increased amounts of infections. So some viruses are starting to suggest that potentially they may play a role in the development of celiac disease. How would a viral infection, say, potentially increase your risk of celiac disease? What it does potentially is change the way that T cell behaves when it's first exposed to gluten. So when we diagnose someone with celiac disease in a family, we sometimes get asked, well, okay, well, what's my risk? What do I do to reduce my risk? A nice study looked at people with celiac disease, and they looked at first-degree relatives. So an index case in the situation was diagnosed with celiac disease, and they looked at their family members. And they looked at a whole heap of different studies. They did this through a process called what we call meta-analysis. Where it's basically we get a whole heap of studies that are independent, and then using a statistical method, combine them all to prove the significance of the results, or increase, sorry, the significance of the results. And they looked at the rates of celiac disease in first-degree family members of people with celiac disease. There was seroprevalence, so positive celiac serology, amongst the first degree family members was 11%. And the actual rates of biopsy confirmed celiac disease was 7%. So if you consider that and compare that back to what we talked about initially, with rates of 1.4 and 0.7% respectively in the general population, you can see that certainly there's a much higher risk amongst first degree family members. Again, we saw that the prevalence was higher in females than males, so 23% of daughters, 14% of sisters, as opposed to 6 and 9% of sons and brothers. Some of the general rates, so if you're a parent of someone with celiac disease who's diagnosed, as a parent, your risk is about 1 in 25. As a son or daughter of someone with celiac disease, your risk is about 1 in 11. And as a sibling of someone with celiac disease, your risk is about 1 in 10. There are slight differences, obviously, depending on male or female sex. They also looked at symptoms, and interestingly, again, 34% of first-degree relatives were with biopsy-proven celiac disease were asymptomatic, so they had no symptoms of diagnosis. Important just when you think about screening. Symptomatic, when you talk about symptomatic patients, there are both gastrointestinal and non-gastrointestinal symptoms. So again, we focus on the GI somewhat, but there's also all those non-GI symptoms that you can see. Not to spend too much time on it, but you see the common symptoms like abdominal pain, bloating, diarrhea, constipation, etc. But then some people develop reflux and other symptoms like that. And also when you look at the non-GI symptoms, there's a wide spectrum of symptoms. People can have significant musculoskeletal symptoms, whether it's arthralgias, can have issues with growth. Also, there are issues potentially with poor energy levels, lethargy, etc., due to iron deficiency. We did look at some local data, and this was performed through the Wesley Research Institute, where we looked at people with celiac disease and looked at the first-degree relatives and then screened them. So we looked at 202 first-degree relatives of patients with celiac disease. Of those, 86% had a positive haplotype, positive HLA. 16 had positive serology, and of those, 10 were children, 6 were adults. 7 of the 10 children ended up having an endoscopy biopsy, and so they had celiac disease. So a prevalence of about 11%. 2 of the 140 adults, first-degree relatives, had biopsy-confirmed celiac disease, so a rate of about 1.4%. Sometimes you get asked, well, okay, I've had a negative blood test. I've been negative blood test for celiac disease. Should I be tested again? Well, how often should I be tested again? If I'm tested now and I'm negative, will I ever develop it? So it's a difficult question to answer, but certainly there's good evidence showing that you should have follow-up testing and follow-up screening. Particularly in pediatrics, we like to retest people because when you're a younger child going through adolescence, it's your peak time for growth, for laying down your bone density. So it's important that we don't miss someone with celiac disease. So we normally would test someone every 4 years if they're a first-degree relative of someone with celiac disease. There was some nice data that came from Northern Europe, from Finland, where they looked at 600 people who were predominantly first-degree relatives of patients who have been diagnosed with celiac disease. Initially, these people had been screened and they had a negative blood test. They then followed them up over time. And there was about 11 years between their screening. And 2.5% or 15 developed celiac disease. Roughly half in the first 5 years and the following half at the final screening at 10 to 11 years. So just saying it's important that if people are tested, they test negative, they do get followed up. So celiac disease is a systemic disease, and just kind of highlighting that fact that a number of different organ systems are involved, and that obviously has important implications with respect to follow-up, but also to potential complications. So in pediatrics, growth is quite common to be impaired at diagnosis, around 10 to 40%, and it's probably the most common extraintestinal manifestation we would see. Short stature and growth delay, so established growth impairment, is only about 10%. It's quite interesting because it's quite multifactorial. It's not just, well, my small bowel is damaged, so I'm not absorbing nutrients properly, so then because of that, I'm not growing. There certainly is an effect directly on the endocrine axis. So what I mean by that is normally your brain releases a compound called growth hormone, which then stimulates your liver to create another compound called IGF-1. And what IGF-1 does, it makes things grow. So it makes your long bones grow, it makes your muscles grow, etc. And it's shown quite nicely that some of the cytokines which are released in celiac disease, they actually block the way that growth hormone works. And that directly affects the growth. We also know that in celiac disease, there's altered bone mass and bone metabolism, which we'll talk about in a second. So thankfully, though, with pediatric celiac disease, if you are diagnosed with celiac disease as a child before you've completed puberty, you should have catch-up growth. So when you finish puberty, you'll stop growing, basically. So if we miss someone as they've had celiac disease that's active throughout puberty and they complete puberty and we haven't diagnosed them, certainly they will not attain their final adult height or their true genetic potential. But certainly when we see someone and we put them on a gluten-free diet and they commence a gluten-free diet, they will see catch-up growth. Typically, we see weight gain first of all, and then usually within about 12 to 18 months post, we'll see that kind of really catch up in growth. As I mentioned, bone mineral density is certainly impaired at diagnosis, regardless of the presentation. There's an increased fracture risk. Again, it's related not only to nutrition, but also to cytokines and some other processes. Up to 75% of people will have osteopenia or weak bones. Thankfully, again, with children, when they're diagnosed with celiac disease, most likely the bone density will be reduced compared to their peers. But if they start a gluten-free diet, within roughly 12 months, as long as they've got a strict with a gluten-free diet and they've got a well-balanced, nutritionally replete diet, their bone density will normalize. Obviously, if they're non-compliant into the future, and obviously this holds with adults as well, bone density will be impaired. And we know that in adults, if you have persistent villus atrophy or persistent damage to your small bowel, there's certainly of hip fracture. Puberty can certainly be delayed also. About 11 to 20% of celiac patients in some reviews, not only just nutritional, but again, issues with cytokines and some autoantibodies that are floating around the bloodstream. And thankfully, though, again, when celiac disease is recognized, usually there will be recovery and people will complete puberty normally within 6 to 8 months of commencement of a gluten-free diet. Again, highlighting it's very important that they're adherent to the gluten-free diet and they have a well-balanced diet for that to occur. The reason I mention this as well is that it carries through into adulthood. Obviously, if you've got active celiac disease, there is an effect on reproductive health. So we know that fertility can be impaired. There's an increased risk of miscarriage. Also, intrauterine growth retardation for babies, so babies that come out smaller than they should be, and low birth weight. And there's also an increased risk of preterm birth. And this is all coming about from unrecognized or poorly treated or managed celiac disease. Autoimmune disease, celiac disease is an autoimmune condition, so it does follow that there's an increased risk of other autoimmune conditions. We know that autoimmune disease in general is increased in celiac disease. So thyroid disease, the general population risk is about 0.5%. If you have celiac disease, your risk increases to about 4%. So there's still over a 90% chance you won't develop it, but certainly it's the most common autoimmune disease we would see in celiac disease. When it comes to type 1 diabetes, usually it goes the other way, in that people are diagnosed with type 1 diabetes and then develop celiac disease. So around 13% of type 1 diabetics will develop celiac disease. So thankfully, it's not as common for it to go the other way. So we do see it though. With celiac disease developing type 1, we'd say it's normally within 5 years of diagnosis and before the age of 20. The risk varies between reports, but it's a little bit less than 2%. This is adult data, and as a pediatric gastroenterologist, I'm probably going a little bit beyond my realm, but this shows you nicely the importance of longer term compliance, but also if you have unrecognized celiac disease. So it looks at people with celiac disease who have persistent villus atrophy, as in persistent damage to their bowel. These are all adults, just to highlight that fact. And they compare them to adults who have villus healing, as in there are people with celiac disease who are in remission. And they look at rates of various complications. The ones in red are the significant ones. And you can see they have persistently low bone density. They have a persistently increased risk of fracture. They have a slightly increased risk of mortality, and also a risk of some malignancies, which are gastrointestinal specific. Just to mention the fact, though, that the absolute risk when compared to the general population is still relatively low. So these cancers aren't common, but your risk is certainly increased as a celiac if you have ongoing villus atrophy and ongoing damage and non-compliance with a gluten-free diet. One thing we talk about all these different medical things, but probably we don't recognize or mention enough is the burden of celiac disease. So if you think about it, someone who's had chronic long-standing symptoms, that has significant impairment in quality of life, but also as well, there's a loss of enjoyment, but also as well, if they've had chronic symptoms, they can have impairment at work, school, what have you. And also as well, if they're not recognized, not diagnosed, then there's risk factors for long-term complications and long-term disease. Post-diagnosis as well, there's obviously following a gluten-free diet is not easy. Also, there's a cost involved in following a gluten-free diet, and also, say for children, separation from peer group, where they have to be careful about what they're eating and whether they can eat out with their friends or not. And also as well, the anxiety of, oh, I'm eating out, am I going to get contaminated? Am I going to get symptomatic? And also, obviously, the economic cost of actually paying for a gluten-free diet. And it does lead to societal costs as well in the longer term with respect to productivity, work, etc. Just quickly, in the interest of time, so I'm probably running a little bit over. Response to a gluten-free diet. In pediatrics, we expect a pretty rapid response. So clinically, people feel better within 2 to 4 weeks. We expect to see quite rapid rates of healing. Said here 95% healing in 2 years, but we've seen kids heal within 9 to 12 months. So they heal quite quickly as opposed to adults who take a little bit longer. We usually follow people up with serology, blood tests, and over time, we'd expect those markers, which are used for, which are initially, high at diagnosis, should halve in 6 months, and then certainly normalize by about 12 months. The vast majority should normalize by 2 years. Obviously, if we don't see that kind of response, then we would consider more intervention about whether we need to do an endoscopy, further intervention. We'll quickly skip this slide in the interest of time. But basically what we normally would do with follow-up, it's an important thing. So it's worthwhile that people have an annual review. Basically, we would normally check the height and weight. This is more from a pediatric perspective, but look at their blood tests, make sure they're normalizing their serology, look for any micronutrient deficiency. Also, we look at those people who say are not responding. That's the point where we'd consider endoscopy. And it's also the importance for follow-up is also to identify family members who aren't being screened, who aren't being identified. And that's probably one of the key things that we do. Otherwise, in summary, when we're talking about celiac disease, it's a systemic disease. Important to recognize that 1/3 of patients are asymptomatic at diagnosis. Important to recognize the higher risk of celiac disease amongst first-degree relatives, particularly siblings and children. It's important to look at screening and follow-up, and also the complications of a delayed diagnosis and non-compliance with a gluten-free diet, particularly in pediatrics with growth, bone density, and puberty, and in adults when it comes to things like fertility, and obviously we mentioned briefly the risk of malignancy. Overall as well, the one thing that's really important for everyone is quality of life, and that's why it's important that we don't miss any of these people with celiac disease. On that note, I will finish my talk and I will hand back to James.
Thank you, Richard. That's excellent. Thanks for providing that overview for us of pediatric celiac disease. I'll just encourage everyone, if they have questions are starting to come through and I'm keeping an eye on them. But if you want to ask a question, if you can just put in the chat function or the Q&A function, and then after Dr. Wagner's talk, we'll go back to Richard as well and have those questions answered. And so now I'd just like to introduce Dr. Paul Wagner again. He's the CEO of Forte Biosciences based in the United States. They're developing a therapy for celiac disease, which goes through a rigorous process. And we've invited him tonight to give us an update on where they're up to with their prior work. So, Paul, thank you very much for joining us, and I'll hand it over to you.
Excellent. Thank you very much. And I really appreciate the invitation from the Wesley Research Institute for hosting this, and of course, Dr. James Daveson, who I've gotten to know fairly well over the last year or so, and really pleasure to work with him, and I really appreciate the invitation to speak with you all. And of course, thank you all. It is a little bit late in the evening here. I'm sure you've all got other things to be doing, but that you've taken the time here to connect and learn more about celiac disease is really appreciated. And so with that, I thought I'd start off just by talking a bit about who we are at Forte Biosciences, and then talk a bit more about celiac disease. Dr. Muir gave a really nice presentation on celiac disease and somewhat on the biology of celiac disease as well, and I'll provide additional perspective there. And that will lead into a discussion of the current therapies that are in development. Of course, right now there are no therapies available. It's just a gluten-free diet, as many of you are fully aware. But on many companies, including, of course, us, are working on therapies for celiac disease. And that will then tie into our discussion about the compound that we're working on, FV102, and some of the data that we've generated with FV102. So with that, we're a biotechnology company that's based in Dallas, Texas, and we're developing therapies for unmet medical needs, including celiac disease. Our focus is on autoimmune disease. Autoimmune disease, of course, being where your immune system has become dysfunctional. And instead of attacking foreign tissue that comes into your body like bacteria or viruses, it instead attacks healthy tissue. In the case of celiac disease, it attacks your intestinal tissue. We've got a number of great employees working with us, researchers working with us, clinical development with extensive experience that are helping to lead the therapeutic development of FB102. And we've also got a great scientific advisory board and board of directors, all with the aim of bringing FB102 forward, eventually, hopefully, to help you all in need in celiac disease. If you want to learn more about our company, the website is right here, and we also are publicly traded in the stock tickers there as well. But just to go back through and maybe give some additional perspective on celiac disease, of course, celiac disease is an autoimmune disease. It is where once you ingest the gluten, as Dr. Muir had indicated, your immune system responds, but it's not responding directly and attacking the gluten. It instead, as a response to that gluten, begins to attack healthy tissue. In this case, that healthy tissue is the lining of your gut or the epithelium of your gut. The symptoms, as has been discussed, include diarrhea, fatigue, headaches, nausea, diarrhea. And there's really, as you all know, no available therapy for celiac disease. It's just a gluten-free diet. And that is not sufficient for many people with celiac disease. Trace exposure happens quite frequently. Learned recently in a discussion with a celiac patient that even paper straws have gluten in them, and that was a shock to me. So it's very difficult to avoid it, and very frequently, unfortunately, celiac patients get exposed to trace amounts of gluten and have severe quality of life issues fairly regularly. A large number of patients, as has been discussed already, and a lot of work going on here. This is a presentation that was shown to me by Professor Jason Tydin, who's at the Royal Melbourne Hospital. It really helped me understand a breakdown of celiac disease. And if you think of it in terms of symptoms and gut damage, the largest component of celiac disease really is with no symptoms and no significant gut damage. That would be called treated celiac disease. It's about 80% of the total. And their gluten-free diet is typically sufficient, but again, there's that trace exposure, which can have significant quality of life issues. If you instead have symptoms, but no gut damage, even after not having been exposed very recently to gluten, that's considered non-responsive celiac disease. It's about 10 to 20%, and there are two components of that. It's either symptomatic with no gut damage or gut damage without symptoms, but again, not having had a recent exposure to gluten. And so it's a sort of a persistent effect of a historical exposure to gluten. And again, that's about 10 to 20%. And then that last quadrant in the upper right-hand corner is resistant celiac disease. Fairly small, but also very severe. That's where there are symptoms and gut damage after a historical exposure to gluten. And in some cases, both this resistant celiac disease type 1 and type 2 can be quite severe, life-threatening. In fact, 5-year survivals can be about 50% in that. And this is just a cartoon, again, maybe a little bit different perspective than Dr. Muir and reinforcing what he was just talking about. But in this cartoon, those little finger-like projections are the villi, and that is what ends up getting damaged when gluten is ingested. When gluten is ingested, those little cells down in the lower left-hand corner, the antigen-presenting cells, are breaking down that gluten, presenting it to another immune cell that then releases these signaling molecules. In this case, they're called cytokines. So here we have IL-2 being released, and that wakes up another component of the immune system, these CD8 positive T cells, just another component of the immune system. And that's reinforced by IL-15, another signaling molecule. And together, that then results in these cells, these CD8 positive T cells, being the bad actors. They're the ones that are then destroying the epithelium of the gut, and that's the pathology of celiac disease. And there's a lot of work being done right now in the therapeutic development across a number of different pharmaceutical companies to try to address celiac disease. On the left is gluten modification. That's where you try to break down the gluten, and I think it's a great idea. One of the challenges with that is knowing when to use it and how much to use. And again, if it's a trace gluten exposure, you may not even know that you've been exposed, and so it might be too late to take a therapeutic that would break down the gluten because it's already been digested, it's already had the negative effect. Then we have immunotolerance, and that's where these therapies may mimic gluten to try to desensitize your immune system, or to try to tie up that breakdown product of gluten. Some of the challenges with that can also be that the therapy itself can give you significant symptoms and quality of life issues. Maybe it'll be temporary as your immune system desensitizes. So that work is ongoing right now. And then over on the right-hand side is immunomodulatory. That's where companies are looking at trying to identify the components of the immune system that have become dysfunctional and trying to quiet those down specifically. And that's exactly what we're working on with our approach at Forte Biosciences, FB102, which is in the upper right-hand corner there with the CD122 approach. We'll talk about that more in just a minute here. So what is CD122? I'm going to keep the science at a fairly high level here, but essentially it is part of a receptor for those signaling molecules I referenced before, those IL-2. I showed you that cartoon. I talked about the signaling molecules, sort of that cascade of signaling molecules that happens when you ingest the gluten involving IL-2 and IL-15. When that IL-2 and IL-15 interacts with its receptor, it causes those cells to grow and divide, to become activated, which means to release these more signaling molecules and to attack healthy tissue. And so we're blocking that interaction. But importantly, we're not blocking the interaction of the signaling molecules with immune cells that are actually trying to help quiet down the immune system. Cells like regulatory T cells, which is in the upper right-hand corner. So we're trying to be very specific about the cells that we're quieting down, only those cells that are responsible for the damage in celiac disease. And again, I'll keep the science at a high level, but this is in vitro data. In vitro just means that it's done in a petri dish, but we use cells derived from humans. And so these are the bad actors. These are the cells that are typically responsible for damage to the tissues. And what happens here, what we've done is in the red, we've exposed these cells to IL-2. In the blue, we've exposed them to IL-15. What you can see on the left is a specific set of immune cells called T cells. And when they're exposed to the signaling molecules, either IL-2 in red or IL-15 in blue, they grow and divide. That's what those bars are showing, that you see a growth and division of those cells. But when you put FB102 into that mix, that's the solid red and the solid blue bar, they don't. In fact, that gray bar over in the far right are the cells just sitting alone without any signaling molecule whatsoever. So effectively, FB102 is preventing these cells from seeing that signal to grow and divide. So it's helping to quiet them down, exactly what we want to have happen. In the middle is another different set of immune cells that have some implications in celiac disease. But on the right, again, those are the good cells. So those are the cells we don't want to be quieting down, the regulatory T cells. And in that case, with FB102, when these regulatory T cells receive that signal from IL-2, they continue to grow and divide as they normally should. So FB2 is not interfering with that. And we referenced activation. Activation is releasing all of these signaling molecules that wake up the immune system and recruit the immune system and potentially do damage to healthy tissue. And these are a number of these signaling molecules. And what we're showing here, there are 3 different human donors. That's why there's the 3 sets of bars. And again, red is IL-2 and blue is IL-15. What we show is that without FB102 present, that we have a big release of these pro-inflammatory cytokines, these signaling molecules. But the 2 columns to the left, you don't see anything there. That's with FB102 present. And the third column over to each of those sets of bars is the cells only. And what you can take away from this is that with FB102, there is no release or very, very little release of these pro-inflammatory cytokines, these signaling molecules. And so you don't have this fanning out wave of waking up and agitating the immune system to attack healthy tissues. This is exactly what we wanted to see in vitro. But now moving from in vitro, we want to move over to, well, what is happening in humans? What human work have we done? To do that, I want to talk a little bit about how we measure celiac disease in humans. And so I showed you that cartoon before. Those finger-like projections are called the villi. That's where your nutrients are passing over those villi, and that's where the absorption occurs. Well, this is actually an image, a microscopic image from a patient from one of our studies with a cross-section of one of those villi. That's the finger-like projection. Those arrows there are pointing to the green dots, if you can see those, and those green dots are those bad actor cells. Those are the cells that are attacking the villi and causing destruction of those villi. And we call those interepithelial lymphocytes. That's just a name for these bad actor cells. What's important here is that once that gluten is ingested, there's an increased infiltration of those cells into the villi. They rush into that villi and they start to attack it. And in fact, you can count them. And if on exposure to gluten, there's an increase of 30% or more of those cells in the villi, that's considered clinically significant. In other words, you now have enough of those cells in the villi that you're going to get destruction of that villi, and it's going to cause damage to your intestines and eventually also cause some of the symptoms that you're all too familiar with. Another parameter we look at is called the villus height to crypt depth ratio. Again, this is exactly the same thing that I showed before with that cartoon. These are those finger-like projections, the villi. In this case, again, we're just looking at that ratio of those two green lines. And what happens when the IELs, those interepithelial lymphocytes, the bad actors, infiltrate into the villi, they destroy it, and so they get shortened. In other words, that VH:CD ratio goes down. And what you want to do when you're developing a new therapy like ours is to prevent, number one, those interepithelial lymphocytes, those bad actors, from infiltrating into the villi. And number two, you want to prevent the villi from being destroyed. And that in itself should begin to affect the celiac disease. Now, there's what's called a composite endpoint. That just means we're taking those two different parameters and we're putting them together so we get a more holistic view of how a therapeutic is working. So with that, this is a study that we have completed, and I'll show you some of the data. We're really excited about this. We presented this publicly at the end of June, so fairly recently. But this was called a phase 1B clinical study. If you're not familiar with clinical development, phase 1 is healthy volunteers, typically looking at safety and tolerability. Phase 1B into phase 2, you're looking at different doses that may work, looking at early signs of activity. And then as you get into phase 3 development, you're more rigorously looking at the safety profile as well as the activity, which then gets submitted to regulatory agencies for approval and then can be used by patients like yourselves. So fairly early, but still, I think, very compelling results as we continue to move this program forward. Here we had a 3-to-1 randomization, which means of 4 subjects that get enrolled, 3 would be on active drug, 1 on placebo. They went through 4 doses of FB102, and then there was a gluten challenge. So we actually stimulated with gluten to see what the effect would be, and we measured the histology. Those slides that I was showing you before, we did biopsies and then looked at the histology to look at those IELs and that VH:CD ratio. And we also looked at symptoms in this study. Baseline demographics are important. Again, we wanted to make sure that these were balanced, very consistent with the overall population that Dr. Muir had described previously as well. So in terms of the activity, this is the data that we showed. This is that composite endpoint. Again, this is the holistic view of combining together those immune cells, those bad actors, as well as the damage to the villi. And here what we showed is a highly statistically significant result showing that on placebo, there was significant damage being done. But on FB102, little to no damage was done at all. In fact, maybe even slightly positive there. Now, when I say highly statistically significant, what I mean by that is that it's highly unlikely due to chance that we're seeing these results. In other words, more likely that the drug is having some therapeutic effect. That's just what that probability means. And the p-value below 0.01 is highly statistically significant. Now, this, I think, is a very important slide in that what we're talking about here are those bad actor cells themselves, these interepithelial lymphocytes, those CD8 positive T cells I showed you before. These are what are causing the damage to the villi. If you remember what I said then, if a 30% increase after exposure to gluten is seen, that's considered clinically significant. Clinically significant, meaning that at that level of IEL infiltration, you're going to see damage to the villi typically. So what did we see here? Well, on placebo, when we stimulated these patients with gluten, there was a 13-point increase. Just remembering back to that baseline demographic slide I showed, as a baseline of 25. So this equates to about a 50% increase, well above that 30% cutoff I just referenced. But if you look at the active arm, that's the FB102, actually saw a decrease. Now, this is after being stimulated pretty aggressively with gluten. That gluten challenge that we used was 8 grams per day for 2 weeks. A typical healthy diet would be about 11 grams. So it was a fairly large amount, maybe the equivalent of 2 to 3 slices of wheat bread. And with that stimulation, actually saw an improvement. Saw those IELs go down, not up. And so we're really encouraged by these results. Highly statistically significant. Here you can see that p-value of 0.0035, so unlikely that these are due to chance. And then the nice trend in the VHCD, it was a short gluten challenge. The damage to the villi typically happens over time. It's a little bit more variable at shorter time points, but as you have those increased IELs for a longer period of time, you'd expect to see more damage. But certainly a strong trend here with that villus height to crypt depth ratio at the end of the study. And what's important as well in this study, we didn't just look at that histology. We also looked at symptoms. And in this case, we were looking at the gastrointestinal symptoms, the GI symptoms, which you're all very familiar with. That's the vomiting, the nausea, diarrhea, abdominal pain, abdominal bloating. And what we observed in the study is that during the 16-day gluten challenge, we actually saw about a 40% symptom benefit, 42% symptom benefit for FB102 relative to placebo. And what's important if we break this down a bit further is that there were a significant fraction that had no symptoms whatsoever. About 30% had no symptoms on active drug. And if we take those that had no symptoms out, those that did have symptoms, there were still fewer symptoms for those on active versus placebo and less severe symptoms. So all of that, I think, was encouraging. We're going to continue to investigate this as we move the program forward. We understand how important symptoms are to all of you, in addition to the potential for protecting your gut, the gut damage. On the safety profile, again, this is an early study. One of the key focuses of this study was on safety and tolerability. And what we showed here is that FB102 appeared to be fairly generally safe and well tolerated. Only one severe adverse event, but that was on placebo. And that was an individual who, during the gluten challenge, had been exposed to the gluten and had a severe reaction to it. They had nausea and vomiting as a result of that exposure. But if we even dig down a bit deeper into the safety profile, again, very favorable for FB102. So we're encouraged by these results, and we continue to move this program forward. This was a study, by the way, that was conducted entirely in Australia and New Zealand. There's just a wonderful group of engaged patients like yourselves who are very, very appreciative of that, and a great infrastructure sites, for example, like Dr. Daveson's, and we've appreciated his involvement in the study as well. And that has allowed us to move forward into a phase 2 study. This also is ongoing in Australia and New Zealand. The details of this, if you want to see them, that's all publicly available on a website called clinicaltrials.gov. It's a site that posts publicly clinical trial activity. So all of these details are there, and this is just a schematic of the study that's up and running now. But here it's a bit bigger. We're going to have, rather than 32 subjects, as we had before, and we're now into phase 2. So now we're looking at different doses, and we're going to have 100 subjects that are enrolled. It's going to be 2 to 1 randomized. What that means is that out of 5 patients, 2 will go on to one set of active doses, 2 will go on to the other set of active doses, and 1 will go on to placebo. So a good probability of getting onto active drug. And you can see here a little different as well. We'll have a little bit more of the FB102. Before it was just 4 doses. Here you're going to get 5 doses up front, which is in induction weekly. And then we're going to go to a maintenance or every other week dosing at either 3 milligrams per kilogram or 5 milligrams per kilogram. And there'll be a bit longer gluten challenge. As you saw before, that VHCD ratio was trending in the right direction. We had that high IELs on the placebo. We would expect that with a longer gluten challenge, we'll see greater differentiation there. And that's going to be important for the regulators to see that in order for us to continue to move this program forward. But we're really encouraged, and certainly couldn't do it without the support of patients. So really appreciative of everybody who's participated in the previous study and this one as well. So with that, I will turn it back over to Dr. Daveson for questions and answers.
Thank you very much. Thank you, Paul. That was excellent. An excellent review. I think that's one of the benefits of these webinars is that we're getting access to what is really very early data. And Paul, can I just ask you, so thank you for presenting your phase 1 data. Can you just explain to the people listening tonight what you would now do with that data. Are you going to present it again internationally? Then is it publication and peer review? What's the process from here?
Absolutely. It's really important that that this ends up being peer reviewed. It was a rigorously run clinical trial. It was a double-blind, placebo-controlled trial, which means that the double-blind portion means that nobody knew who was on drug, who was on placebo, and that helps to keep us all honest. But part of it is awareness. We want people to be aware of this drug, and so we certainly want to be able to present this to a broader audience of clinicians and eventually to the patients as well. So we're actually scheduled to be presenting this data. Professor Tydin will be presenting this data at the Tampere Celiac Disease Symposium, September 10th through the 12th. We'll have a little bit more data than what I presented here, again, supportive of the clinical trial. And then we hope to publish it in a peer-reviewed journal, and we would hope that happens by the end of the year. So if you want to read more about this in a scientific journal, medical scientific journal, we should hopefully have that available before the end of the year.
Yeah, thank you. Well, there's a couple of questions that have come through for you, actually. So we might just start with those, if that's okay, before we head back to Richard. So the first question was, which I'm sure you're able to answer. So for people in the phase 1 study that had celiac disease, were they treated or was their disease successfully into enrollment?
That really was our focus. Our focus was on those subjects, those patients who had were on a gluten-free diet. And that would mean that they've got a relatively normal VH to CD ratio. That normal ratio should be in the 2 to 3 range. I think we actually did allow down to about 1.5, which would be maybe on the edge of showing some damage, but fairly asymptomatic. They should not have had a gluten exposure, significant gluten exposure over the last 12 months. And there was a clinical diagnosis of celiac disease. So we had the paperwork to show that they had been diagnosed with celiac disease. And part of that was to show that once they were exposed to the gluten, that as I was just demonstrating with the data, when they're exposed to gluten on the placebo, that there was a significant response, both in terms of symptoms and histology. Whereas when they're on Epi-102, what we were hoping to see, and we were fortunate enough to see, was that we were able to block some of that damage on the histology as well as mitigate some of the symptoms.
Yeah, great. And just to clarify that for people listening, in case they don't know, when Dr. Wagner is talking about VHCD, as he pointed out, that's the height of those villi compared to the crypts that lead into them or are sitting next to them. So 3:1 is probably normal. He was just mentioning that some people, 1.5 to 2 is probably what most people would have.
And I'll just say too, our intention is with your feedback and with regulatory feedback is to continue to develop for this the subjects with that are on a gluten-free diet well controlled, in part because we've heard from many patients that even though they have worked hard to stay well controlled and avoid gluten, it's very hard to avoid gluten. I've had stories about patients who have had sandwiches made on panini makers that had crumbs of gluten-containing bread on them and ended up getting sick from that soy sauce that contained it. Again, as I said, I learned just last week or earlier this week, I guess it was, that the straws, paper straws, have gluten in them, and it's very difficult to avoid it. So if there's a way to prevent yourself from ending up with some severe quality of life issues by taking a therapeutic like this, we'd like to be able to help you out and to be able to provide that. And so that's our intention. I do also think this therapy could very well work, as I showed you in that quadrant, those grids, the non-responsive and maybe even in resistant. But we'd really like to be able to take this forward in that gluten-free, the treated population, the patients on a gluten-free diet.
Yeah, thank you very much, Paul. I might move on to Richard and just throw him a couple of questions and give you a break for a minute. So Richard, there's a lot of questions coming forward for you. One was, can you just go over again your 4-yearly testing strategy? So when do you start testing high-risk kids for celiac disease? If they're negative, how often do you do it? And do you incorporate gene testing early on?
Yeah, I mean, certainly, normally we'd screen every 4 years in pediatrics. Sometimes we'd focus a little bit closer with respect to around that time, depending on how old the child is, particularly around that time of puberty, because we don't want to miss people. Certainly, initially for a child who is a sibling of someone with celiac disease, I would normally do the gene test initially. So the haplotype is negative, then we can say, well, okay, you're not going to develop celiac disease. We don't need to test you again. Obviously, if haplotype positive, then obviously we'd look at follow-up screening into the future. But normally I would say every 4 years or so, yes. As in the adult population, how often would you test your patients?
Well, celiac disease, you're throwing it back at me already.
I have, sorry, but I just thought it'd be interesting for everyone who's listening just to think, well, okay, we've talked about family members, what happens in the adult population?
Yeah, no, I think it's a good question. So I'm always very interested in the HLA typing, and if that's negative, that obviously I think pretty much excludes celiac disease in first degree relatives, which are the highest risk group, and that's where we're trying to find cases of people with celiac disease. I think it's important that Richard pointed out that in his study that he did, 14% of first degree relatives had undiagnosed celiac disease in children here in Brisbane, where I think he was saying it should have been 7%. So there's a lot of kids that are undiagnosed with celiac disease in Australia at the moment. So having a heightened awareness in those high risk families of people with disease is really important. I wouldn't necessarily test them all the time. I would retest them when they're unwell, or maybe every couple of years when they're having blood tests for another reason for their celiac serology. What I do see sometimes is people who don't have celiac disease, they may go overseas, they might get a viral infection, they might become unwell. That's when I would retest them. Sometimes viruses can promote immune activation and lead to development of autoimmune diseases. So that's the time when I'm particularly interested. Richard, another question on that theme. Second degree relatives, so nieces and nephews of people with celiac disease, considered high risk for the development of celiac disease.
So the risk does drop somewhat. So certainly amongst second degree relatives. So certainly current recommendations are we don't normally screen second degree relatives, but certainly, I mean, at the end of the day, if there's a clinical concern, obviously there's clinical symptoms, it's quite reasonable to, but at the moment don't typically recommend screening second degree relatives.
Yeah. A common question or a common theme that's coming through, I think, is what age do you recommend introducing gluten to children of people with celiac disease? It's a common question, but yeah.
Sorry if there's any confusion about that. I mean, I certainly wouldn't necessarily delay the introduction of gluten at all. The data is more based around when it is introduced, it's introduced at significantly high amounts. So it is safe to certainly introduce gluten as you normally would with introduction of solids. And there's some previous good long-term data showing that, say, if someone is at risk of celiac disease as a first-degree relative, if you delay the introduction of gluten, you don't necessarily reduce the long-term risk of development of celiac disease. That basically, if these people were going to develop it, they will develop it. They looked at early and late introduction of gluten in high-risk patients. And certainly, initially, there's a slightly higher risk than those patients who were introduced early at 2 years of age. But by 5 years of age, there was no difference between the two groups, those that had early or late introduction of gluten. So delaying it's not going to make any issue and not going to make any long-term difference. Certainly, though, when you would introduce it at a normal time, but just saying that the data was like, a slice of bread for like, as I said, an 8 to 12-month-old, which is a lot to have every day. And that was more in Europe, Northern Europe, which showed that there was a slightly increased risk. So I wouldn't delay introduction of gluten. It's still safe to introduce gluten as you would normally. Certainly you can just do it regularly. If you want to pursue a slightly lower gluten introduction, that's fine. The problem at the moment is we don't have any formal guidelines from any of the paediatric societies or otherwise to say, well, okay, this amount is safe, that amount is not safe.
Okay. Paul, I just might go back to you. From the concept of an idea to a treatment for celiac disease to actually delivering it to people with celiac disease, it's often quoted it's around 10 to 15 years for that process to occur as it goes through the different phases of studies. Could you give us an idea of how long you've been or your team's been working on this, where you're up to in that timeline?
Absolutely. There's two components to that, really. It's the time and the cost. As a public biotech company, part of what we do is to raise capital to try to fund the program to continue to move it forward. And those two are intimately related. Our investors are expecting that we move this forward as quickly as possible. Of course, we want to do that to be able to help patients. This program, if you start from the very, very beginning of it, where we first made this molecule, started about 4 years ago, really. And so we made the molecule. We were doing a lot of early testing with it in the lab. That was at the research stage. Then we moved it into animals. This is the typical progress of drug development. We moved it into animals, mice first, and then non-human primates. And then from there, we did our first healthy volunteer study, which was just over a year and a half ago. So we did do a healthy volunteer study. That was a phase 1 that I described before, which we're really looking at safety and tolerability. Then we moved it into the phase 1b, where we went into the celiac patients, which is the data I just showed you. And now we're in phase 2. There's hopefully going to be a good response to this study in terms of the enrollment, and that we'll be able to enroll it quickly. We'll be able to read it out fairly quickly. Next year is currently our expectation when we'd be able to read this out. And then after that, we'd have a discussion with the regulators. There's guidance from FDA about what they're wanting to see for drug development in celiac disease. We're hopeful we can broaden that a bit from where their current focus is, but that I think we could be in a phase 3 study shortly after we read out the phase 2 study. And then after that, we'd be able to submit that phase 3, if positive, to the regulators for approval. I would say that would probably be in a 2028-ish time frame. I mean, that's if everything goes really, really well for us, that would be sort of the time frame. So not that distant future, maybe 28, 29, not too far in the future that a therapy could be available. It is very important, though, as long as that is, it's really, really important that we have that level of rigor. Large clinical trials to show safety and activity to keep patients safe and make sure that this is a drug that really, really does work. It's also very expensive. So along that way, not only is the clinical trial failure risk, we will need to make sure that we continue to get funding. The typical number that gets thrown around, and this might be surprising to some of you, the number that gets thrown around for the cost of development is about $1 billion. That takes into account that from the very beginning all the way through to approval, you only have about a 10% success rate for a typical therapeutic. So very low success rate, very expensive, and it takes a long time. But it certainly is worth it to be able to bring a drug to help patients in an unmet need like celiac disease.
Yeah, thanks, Paul. I think that's a really important message for people with celiac disease, that the chance of these drugs making it to the market is actually low, but there are a lot of them being trialled at the moment. Just with the side effects, Paul, I think in your phase 1b study, could you just go over that again? There's been a couple of questions about the side effects you'd anticipate or what you've seen.
Yeah, that's an excellent question. I think one of the nice things about the type of molecule we're developing is that it's very, very specific. There are other therapies that are broader on their impact to the immune system, things called JAK inhibitors that kind of shut down broadly the immune system and have black box warnings, which is a warning that's at the very beginning of a label that, there's a lot of safety problems with that compound and you need to be aware of it. That's what a black box warning is. And it knocks down those regulatory T cells that I said, we want to be careful to protect those. So because we're very specific in interfering with the signaling of those cells that have become dysfunctional, it really is those dysfunctional cells that we're impacting. In a typical healthy individual, you don't see significantly elevated levels of IL-2 or IL-15 for that matter. And so blocking that pathway doesn't have that much of an effect because there just isn't that much IL-2 or IL-15 around. Unlike in celiac disease, actually, there's some nice publications. Professor Tydin has one of them showing that when you stimulate with gluten, without any therapeutic intervention, you have a big spike of IL-2 that happens right after the exposure to the gluten. And so when that dysfunction happens, that's where our therapy then has an effect because we block the downstream impact of that elevated level of IL-2. If you don't have elevated IL-2, we shouldn't really see too much of an effect of a therapeutic like ours. Now, again, it's early days. I don't want to say that there's no safety components we're focused on. Of course, that's rigorously what we're looking for. That was what the phase 1 study was about. That was what the 1b is. Safety is a very important part of this all the way through, but so far it looks generally safe and well tolerated, and based on the mechanism, we're hopeful that continues.
Yeah, thank you very much. Richard, I'm not sure how much more time we have for questions, but I might just pop another one to Richard. Occasionally, I think the presentations drop out, but there's been a couple of questions about the premature babies and the risk of celiac disease. Would you mind just clarifying that again for us?
I mean, there's certainly a slightly increased risk in that, there's a risk there seems to be an effect with active celiac disease on placental health. And as a result, that increases the risk of, as I said, intrauterine growth retardation, low birth weight, and certainly preterm birth, yes. So the risk is increased. Obviously, there are other risk factors for premature babies as well, though. But certainly, yes, there's an increased risk from a celiac disease.
And do you see much, there's questions about other allergies or allergic diseases. Do you see much peanut allergy in children that have celiac disease? Are the two coexist in your practice?
No, not typically. We wouldn't say it's an association. It's a slightly kind of different IgE-mediated process. So no, wouldn't say that's typically something we associate with celiac disease, no.
Very good. I think we might have to think about wrapping it up. And I think we've covered most of the themes. So I'd just like to take the opportunity now just to again thank both Dr. Muir and Dr. Wagner for their excellent presentations and for taking the time to spend with us tonight to update us on clinical aspects of paediatric celiac disease and also really what is cutting edge in terms of therapeutic advances for potential therapies for celiac disease. There's been a lot of thanks coming through as well. So again, thank you very much.
Thank you. Really
Thank you very much. Cheers.
So Wesley, just to finalise, thank you all for attending today. We're really grateful that you've all joined us. Wesley Research Institute is a not-for-profit. You can join either by looking on their website. You can participate in a clinical trial. We'd love to have people participate in clinical trials. Or you can join the Celiac Research Network and you'll be updated with webinars and any other events that we host here at Wesley Research Institute. Sorry, this is the last webinar we had, so sorry for the mugshot, but this will be posted. This webinar will be posted on YouTube, and when you get to Paul's presentation, it's a bit clearer to understand. Thank you very much, everyone, again, and we look forward to you joining us for the next one.